So I don't see the screen anymore, but I guess you can still hear and see me. So let me then introduce the next speaker, which is Professor Hannes Mikula. After finishing his studies in technical chemistry, Professor Mikula obtained his PhD from the Technical University in Wien, in Vienna, in 2014, where he was already inspired by the field of bioorthogonal chemistry. And that made him join the group of Professor Ralph Weisleder at the Massachusetts General Hospital and Harvard Medical School as a postdoc, where he spent 2 years. And I think that really also shaped his ideas for his independent career. In two years later, he returned to Vienna, where he started as an independent young principal investigator to lead the group on molecular chemistry and chemical biology at the Institute of Applied Synthetic Chemistry. His recent work mainly focuses on the development of bioorthogonal tools and reactions for in vivo chemistry, diagnostic tools, medical imaging, and theranostic applications. Professor Mikula received quite some awards, I would say in the past 10 years or so. Let me just mention a few recent ones. In the past year, the START Award on the Austrian Science Fund, a Science Award from the Austrian Chemical Society and Austrian Academy of Sciences, a Young Scientist Award from the Federal Ministry of Education, the Karl Schlegel Award from the Austrian Academy of Sciences, and also the Habilitationsprize from the Austrian Chemical Society. And there are actually many more, but I think this will do for now. And I gladly would like to give the floor to Professor Mikula to present on, well, I guess, reactions of tetracenes and cyclooctenes. Thanks. Yeah. So the floor is yours, Hannes.
Many thanks, Floris. I hope you can hear me well. Thanks again for the nice introduction. Certainly no need to mention any award, I should say. So it's all about the science today. No, really, thanks again for the kind words. Also, many thanks to Thieme for this exciting opportunity to, let's say, being able to share some of our recent results of our molecular designs. And that's actually what I'm trying to do now. And I guess that works. Always the two standard questions in webinars and online meetings. Can you hear me and can you see my screen?
Yeah, it's all perfectly fine.
Perfect, thank you. Okay, and as already mentioned, and there will be some overlap with Professor Rutja's talk before, also some historical ones, I should say. I'd like to talk about tetracene transcyclooctene chemistry in particular. And the title has been mentioned before. It's all about bioorthogonal bond-cleavage reactions, a story that I usually call simply exit the cube. And you will see why this is sort of my favorite phrase to call our research in the past few years. So as mentioned, I've actually been trained as a synthetic chemist, also sugar chemistry, carbohydrate chemistry. So this is something like this is basically what most people have in mind when thinking about synthetic organic chemistry. And our main motivation right now is really to design reactions that work in vivo. So bioorthogonal reactions, bioorthogonal chemistry, or to be actually more accurate, in vivo chemistry. So when comparing these two, right? So in synthetic chemistry, just thinking about all the crazy work out there when it comes to total synthesis, natural product synthesis. It's always really about one specific chemical structure. And the aim is to prepare this no matter how it can be achieved. So we would have all crazy different manifold methods. And this is actually completely different in the field of in vivo chemistry, at least still completely different, where the structure is not that important, let's say. It's actually the how, as we really aim to have reactions that can safely be performed and that safely proceed and selectively proceed in living systems. So it needs to be biocompatible, highly selective, and so on. What's really important to me, and I keep repeating myself in several talks, is reaction kinetics. So those reactions really need to be ultra fast. And I will get back to this in a minute. So why are we actually interested in vivo chemistry? So it's like a standard phrase. So we want to understand biological processes or living systems, but also manipulate living systems. So that basically means it comes to diagnostics and therapeutics. So the design of new approaches, for example, in vivo medical imaging, field that I'm not going to cover today, targeted drug delivery. So that was already shown in Professor Rutje's slides. So the cleavable antibody drug conjugate will come back soon. Or just briefly calling it, let's say, the on-off control. So can we turn something on? Can we turn something off? And to achieve that, we mainly focus on the design of chemical tools. So similar as Professor Rutjes showed before, I also included a few slides basically showing how I got into this field. And it was a very similar story, although a few years later. So back in 2007, I was still studying technical chemistry, and I just finished, I remember that very well, a course on asymmetric synthesis. And being able to attend this tetrahedron anniversary symposium in Berlin, I still remember I was super excited. I mean, the speaker lineup was very impressive, and it also included Barry Sharpless, which was, let's say, one of my heroes also back then, but mainly because of this course, right? So asymmetric synthesis. So I was mainly, right when he entered the stage and he was about to start his talk, I was expecting crazy structures like this, already sort of getting my brain functioning to imagine 3D molecules. And then, so let's say all of a sudden, he basically showed this. I'd say I was not old enough because I did not get the concept at all. So my first thought was mainly, okay, why triazoles? Why is everybody interested in triazoles? That was the only thing I could focus on. Yes, I understood that this is sort of a very convenient or very efficient reaction, but I completely missed the point that it's actually also all about connecting two things via the triazole. So definitely something I still regret. I could have learned much more from this talk. However, a year later, as I said, I basically started research as a carbohydrate chemist. And back then I was able to attend the International Carbohydrate Symposium in Oslo. And I was like, whoa, that was crazy international meeting, many participants. And it started with an award lecture, and that was the Whistler Award. And back then, the award was given to Carolyn Bertozzi. I was like, whoa, International Carbohydrate Symposium, award lecture. So expecting many sugars. And basically one of her first few slides showed this. And I was like, oh, it's back. Now it can be done copper-free. But at this time, fortunately, I was listening carefully enough to really get what this is all about. And it basically totally blew my mind. That was probably the most inspirational talk I've ever attended. And yeah, to cut long things short, it was certainly related to sugar chemistry. So Carolyn Bertozzi showed the, let's say, metabolic engineering of aceto-functionalized sugar precursors into sugar glycan conjugates, so glycans at the outside of the cell, basically proving that the metabolism can, or tolerates the A site, that this is incorporated into glycans, and at different time points treat a developing zebrafish, so growing, let's say, zebrafish with cyclooctines conjugated to a fluorophore, as I said, at different time points, and then later on really showed that the chemistry is basically happening inside the living fish, and that it's possible at a later point of time to really take a closer look on those glycan structures, also in light of the development process of the fish. So that's certainly one of the landmark papers, if not the landmark paper, in the field of bioorthogonal chemistry. And as I said, completely changed my view on this topic. And that was basically when my fascination started. So we've already seen quite some cyclooctines before. I'm bringing several of those back here. So starting from the original designs, there was a lot of work invested, I can only imagine. And I'm really sorry for using the wrong abbreviation here. So it's DBCO in BCN. But the good thing about these two molecules, I'd say, developed by Professor Rutjes and his team, is I would say that these two are still the most commonly used and the most efficient ones, also commercially available, I should mention. Yes, there are some faster cyclooctines, more reactive ones, but in general, we could basically say that the second order rate constant is in the range of 1. Some other examples now that boosted this kind of chemistry up to 10 or even a little higher. But for, let's say, these cyclooctines shown here with the A site, it's in the range of 1 per molar per second. So it's always like this, okay, what does that number mean, and why do reaction kinetics really matter? So just to discuss that on an example, so let's imagine we have this reaction of A plus B, and that gives C, so a classic click reaction. And we would consider different concentrations of A and B, and now want to know, okay, how long does it take to achieve 95% conversion? Just as an example. And we would go with the rate of 1, then you can clearly see that in a micromolar or even sub-micromolar range, which is basically the range that we need to work with under in vivo conditions to keep the chemistry safe, then it's obvious that this would take certainly forever, especially when the application is, let's say, a time critical one. And as we move up here with the second order rate constant, of course, this becomes more and more feasible. In addition, what I also like to mention is that, of course, a living system is simply not a flask. So, for example, you could argue, I don't really care whether it takes 19 seconds or 30 minutes, especially as a synthetic chemist, right? You would usually say, it's nice if it's done in 5 minutes, but if it's done in 30 minutes, it gives me enough time for a coffee. Or I simply go home and leave it overnight or whatever. However, that's simply because that's sort of like a confined system, a flask, constant reaction conditions. And this is certainly not the case for a living system. So if a reaction is not fast enough, that basically also means that, for example, if we have one molecule that is already, let's say, pre-targeted to a certain position in the body, there is a second molecule sort of like coming by, and we have local concentrations, of course. And for the reaction to occur, this basically needs to be fast. Otherwise, it's not like, okay, let's wait half an hour longer and maybe it clicks. No, it might simply pass the other reactant and not react at all. It might get excreted, degraded, metabolized, and so on. So basically, not fast enough would actually mean it's not happening at all. So pharmacokinetics, right, to come up with a more general term, definitely also has an impact on reaction time. And also mentioned, right, the game changer in our field certainly was the tetrasin ligation or the tetrasin transcyclooctene ligation, as we were certainly in desperate need for speed. So that's just briefly a so-called inverse electron demand Diels-Alder reaction. Inverse simply because it was the second one probably discovered. So it's the inverse electron demand Diels-Alder reaction. It forms these dihydropyridine click product. You would usually see it like this. Maybe a different tautomer is drawn. And in figures, it often says, yeah, plus isomers or only one isomer shown, as usually no one really cares what's going on in this system as long as A and B are connected. I mean, that's the purpose of the click reaction here. The IETA reaction basically is actually only the first step here. So the Diels-Alder cycloaddition, that's followed by a retro Diels-Alder. So eliminating nitrogen, finally forming this system. So of course, it was known from the very first paper. So Professor Joseph Fox, who is definitely the inventor, let's say, of this chemistry. And it was known from the beginning on that this system here is prone to tautomerization. So we can have two different tautomers that are sort of in equilibrium also with this, let's say initial click product shown here. And over time with oxygen, especially under physiological conditions, this can then oxidize to an aromatic system. Most importantly, yes, it's this 5, I would say up to 6 orders of magnitude. This reaction is substantially, significantly faster, I'd say, than any other bioorthogonal reaction. So with second order rate constants of 100,000 and even higher. So you can go higher with more strained transcyclooctenes. I'm not going to show these. This is when it becomes tricky with in vivo chemistry because stability, of course, also plays a huge role. So 100,000 is definitely a good number here. And I should have inserted a reference. So we have recently actually shown with our colleagues in Copenhagen that a number of around 50,000 is a very good, let's say, indicator for successful in vivo ligation, even in short timeframes. So the reaction, of course, is biocompatible, highly selective, non-toxic, and so on. So this is what really enabled in vivo chemistry. In addition to the transcyclooctene, of course, the reactivity also depends on the tetrasin. I'm briefly going to talk about this as we need this later. The reactivity of tetrasins usually, or in general, increases depending on the substituent from alkyl to aryl, electron withdrawing or electron poor heteroaryls that I would say have similar reactivity than, as we say, H tetrasins, so monosubstituted tetrasins here. So of course, that means there are plenty of different combinations possible. And I'd say the aryl H here and the bispyridyl tetrasin, these two are definitely the most widely used ones when it comes to very fast tetrasin ligations and especially in vivo chemistry. The nice thing about tetrasins, despite their cumbersome synthesis, is those compounds are usually bright pink, maybe to red colored compounds, which makes working with these substances sometimes really colorful. And that's also an important side note, as I'll get back to this later. Also mentioned already, and that's sort of the main motivation of my talk, is the advancement of tetrasin TCO chemistry to bond cleavage reactions. So as shown already before, so we can react the tetrasin with the trans-cyclooctene, as we later can call it the release TCO, which is modified here in the allylic position. This will undergo a classical tetrasin TCO ligation. And then, depending on the two different routes for tautomerization, as shown before, form different isomers, where one of these two, and that was hypothesized from the very beginning on, when Marc Robillard, the inventor of this chemistry, published about this reaction the first time. So that was in 2013. So one of these two can undergo a 1,4 elimination, finally leading to release of this carbamate moiety. Following elimination of carbon dioxide releases this primary amine, as shown here. And that reaction from the beginning on was termed click to release. There are several others now called click to release, and this is similar with click chemistry, sometimes hard to come up with a very valid term and definition. However, the main motivation for this chemistry from the beginning on, is also already shown in Professor Rutge's slides, and envisioned by Marc Robillard from the beginning on, was to use this chemistry for the design of ligand-drug conjugates. So, for example, antibody-drug conjugates that, as you know, are basically used to shuttle toxic drugs to the site of disease. So, for example, a tumor. And the activation of these would usually depend on, let's say, tumor biology, uptake into cells, enzymes, pH responsive stuff, and so on. And Marc's, who's basically, or actually running his own company, TechWorks Pharmaceuticals, motivation was to introduce a cleavable TCO in between that antibody and the drug, so that at a later point of time, it is possible to use tetrasins, let's say as bioorthogonal scissors, to cleave the drug off. So which would make this system no longer dependent on tumor biology. So it's also possible to use antibodies that, for example, target receptors that do not internalize into cells. And of course, what's very exciting for everybody in the field is that a different approach, let's say, but certainly the same chemistry is used by Chasqui, so a US-based company, since at the end of 2020 or early in 2021, finally in clinical trials. So this chemistry is, let's say, finally happening in humans, which basically marks the beginning of the era of, let's say, clinical translation of bioorthogonal chemistry in general. So the problem with click-to-release chemistry is there is sort of like even more need for speed and efficiency, right? In the field of click chemistry, we are used to say, okay, we have A and B and it's complete conversion and everything is fine. So in this case, it's not only the click kinetics. So these two compounds need to react with each other. So otherwise nothing happens at all. But following the initial click reaction, of course, the release kinetics play a crucial role. And as you will see, the overall yield of this process, is, at least in the beginning, was far away from being 100%. So the cleavage yield or the release efficiency is an important parameter, and of course also bioorthogonality. And the key problem at the beginning was that when we look at different tetrasins and we go up with the reactivity to, let's say, achieve sufficient click rates here in step one, that we drastically go down with the release yield. So when using commonly applied bispyridyl tetrasins, the release at the beginning was, I'd say, only poor, definitely lower than 10%. And that brings me to this graph that I will use also throughout my talk, where we, let's say, put the release yield and the reaction rate of the initial ligation step, and would say, yep, actually where we want to be is up here. So definitely ideally in the range of 10,000 or even higher in the release yield of 90% plus. And when I started working on this, and that goes back to my time as a postdoc, we were actually wondering at the beginning, okay, so why is release not complete, right? Why is it incomplete at all? And at that time, it was like the dimethyl tetrasin, DMT, was sort of the tetrasin of choice to achieve, let's say, around 80% release. And when looking at this reaction, we were wondering, okay, if this forms the initial click product and there is an equilibrium between the two tautomers that we call B1 and B2, after, let's say, quite some time, there should be release only. I should also add here the final tautomerization here, which leads to the aromatic system, is no driving force of the reaction. And hypothesized from the beginning by Marc Robillard was that there is also oxidation going on of this non-releasing tautomer, and that sort of like leads to release yields lower than 100%. So we did this reaction, let's say no oxygen at all, no oxidants or any other stuff, and we were never able to achieve 100%. So we started with quite a number of evaluations, and I'm not going to go through all of these. What we basically learned back in those days is that this reaction basically depends on everything. So which buffer is used, how concentrated the buffer is, and most importantly, also pH. So at physiological pH, as you can see here, we basically found out that the release is sometimes limited to only 20%. And even, right, when we started this reaction at the higher pH, for example, pH 9 or 8, and let it steer, let's say, for a while, and then went back to acidic conditions, we could not improve release. So we were like, okay, something is going on. HPLC or LCMS clearly showed us it is not an oxidation happening, as the mass was always related to B2. So we were wondering, okay, why is B2 forming, and why is it sort of like suddenly becoming a stable tautomer? And the reason is actually that it was not B2, as we were finally able to show. And let's say unravel the reason for incomplete release. It's always hard when drawing something in, let's say, in 2D, as we're all used to do this, because what's actually happening here is that this initial click product and this carbamate functionality, they can undergo a nucleophilic addition and form, as we call it, a tricyclic dead end tautomer. And this one is no longer releasing. Sounds a little weird, or looks a little weird to draw it like this, but when looking at this, let's say in 3D, using model compounds, we can clearly see that this is actually, let's say, not a far distance to achieve this. So knowing of this tricyclic dead end, we basically added it to this reaction scheme as shown here. And when doing this, that was sort of the first time when we noticed, oh yeah, that basically looks like a cube. And this is when we came up with this, let's say, schematic saying like this is the release cube. And we like working with this because the top layer were sort of like the intermediates, where the bottom layer are the final products. We would have the front face here, which let's say is the bad face, the no release face. The face in the back is the release face. And I will get back to this later because some of you might already notice that, let's say, the release cube has a flaw. However, that is, basically when our journey started to, let's say, exit the cube. So I'm not sure whether you are a fan of exit the room games, or during the pandemic, I'd say many board games became popular. I need to admit I'm totally into this. And that's why it was, basically 1 + 1 = 2 that we came up with this phrase. Okay, can we exit the cube? So how can we circumvent any problems and really boost bioorthogonal bond cleavage chemistry? The first very straightforward idea was, can we avoid the formation of the dead end, which was, as I said, straightforward, because when blocking this nucleophilic addition here with a methyl group, that was the idea back then, and it was not surprising, knowing now what's going on. It was not surprising to see, yes, we can definitely achieve complete release, so complete, let's say 99% plus. However, that was with alkyl, so bis alkyl tetracins only. So what we basically achieved is we moved this pink square here, let's say up from, depends on the conditions, from 20 or 80 up to 100, meaning we're still much too slow. So basically next step was like, okay, can we accelerate this reaction somehow? So that basically brings me back to this initial tautomerization process. And we were like, okay, how can we trigger this? And how can we accelerate also the release step? And that goes back to our very first report when we came up with tetracin acids, actually originally motivated by the fact that release was faster under acidic conditions. And later, Sander van Kasteren came up with a scaffold that I need to admit is way more powerful. It's these ammonium functionalized tetracins. And what these, let's say, directing groups basically do is, after or upon the click reaction, these groups direct tautomerization, so to accelerate formation of, as shown here, the releasing tautomer, and moreover, it also enhances the elimination step. However, and this is something that always needs to be considered, we would basically have two different click orientations of the tetracin. So if the acid, as it's shown here, is, let's say, close to the leaving group, it would direct formation of the releasing tautomer and really lead to fast release. Whereas if it clicks the other way around, it would basically favor formation of B2. There might be some equilibrium going on, but the release is definitely only slow. So, and considering this, we have basically termed these two click orientations head to head and head to tail. Need to admit that was basically the best we could come up with. So when speaking of accelerating, we were like, okay, can we use aromatic tetracins? So heterocyclic tetracins. And just quickly switch here. Hope you can still hear me well. It seems to work. So we were basically wondering what's going on with aromatic TZ and why are these not properly working in release experiment. Made these alkyl heteroaryl tetracins and investigated release. And what we could basically see was, a release of around 50%. Yes, with the acid, as it's shown here, it's basically instantaneous release, right? This is what the acid is doing, but then it is still stuck at 50%. And that's basically what I've said before. So it depends on the click orientation. So if the alkyl here is in head-to-head position, then we achieve release. Depending on whether it's a methyl or an acid, it will be fast or medium. And as soon as the aromatic system here is in head-to-head position, then we sort of get stuck at B2. And we could show that this B2 is no longer in equilibrium with the initial click product. So it is really a stable compound that we can isolate and analyzed by NMR. So it's this 50-50 around ratio of click orientation that is a key limitation, as we simply cannot control the click orientation. So we thought, okay, can we outmaneuver click orientation, and came up with the C2 symmetric, yes, it really is C2 symmetric TCO that has these two leaving groups here in allylic position. And with C2 TCO, as we call it, if we would react this with the TZ that has one directing group, it would no longer matter whether it clicks in this direction or in the other orientation. The directing group will always be in head-to-head position to at least one leaving group, which is why we thought, okay, then we should achieve rapid tautomerization, elimination, and basically instantaneous cleavage. It's a omnidirectional cleavage, and I'll get back to this, of course, as we cannot control which of these two molecules is cleaved. So we thought, okay, this could be fast and complete, independent from the click orientation. And long story short, this is exactly what it is. So we could show this for several tetracins that we achieve basically quantitative release in short time. So we'll see an example soon. And I also want to highlight that we basically also included a bispyridal tetracin. So that was, as you can see, a few years ago. And the release here was still poor, which we thought, okay, that would maybe suggest that the tautomerization is not going on at all. So there can't be any wrong tautomer formed, as we would have two different leaving groups. So we took a closer look and basically found out that it is actually tautomerizing. It even forms to some degree, let's say, the right tautomer, but the right tautomer is still not releasing, or we would only achieve poor elimination. And that was sort of an important information back then, and I will get back to this later. The initial problem for C2 TCO actually was that by introduction of the second allylic modification, we created more steric hindrance, which basically slowed down again the initial click process. So we thought, okay, we can also accelerate tetracins by basically putting no substituent there at all. So in H tetracins that were before that or still known as the worst when it comes to release. But in this case, we thought, okay, this should not really matter as long as this R group here is a directing group. So we would still achieve cleavage here. And we used Sander van Kastelen's, let's say, H ammonium tetracin, here shown as the amine. And yes, that was something that worked really nicely. So again, achieved complete release now with a rate of almost 400, or second order rate constant of almost 400 per molar per second. Just to put this in perspective, that means 10 micromolar TCO plus 50 micromolar TC would mean 99% cleavage in 5 minutes. And that's the rate determining step here, is the click reaction. So we cannot sort of delineate the release here, as it's basically instantaneous. But of course, the problem is we have no control with C2 TCO of which of these two balls here is released. So, and that was also not the intention of this study, because what we aimed for are turn-off applications. Right? I've mentioned at the beginning, on-off control. And what we did, for example, to turn off, so we put the C2...
Hannes, I'm sorry to interrupt, but could you are you coming to a close soon?
Yes.
Yeah? Okay, thank you.
Sorry for that. So we put that in between. We labeled cells with this, and later edit the tetracin and could show that we basically cleave off the fluorophore here completely, and it gets dark again. So why would someone do this? Then you might want to look up the concept of multiplex cellular imaging, and there is a very nice study hopefully coming out soon that we're actually very glad to have participated. However, so to finish this up, basically we were still exiting the cube. So we thought, okay, if the TC acid is improving release and the pyridyl TC is improving click, so why not combine this and create a phenolic one? So let's say hydroxypyridyl tetracins, and our hypothesis was that this acidic group also controls tautomerization and later on helps with the elimination. So Martin, back then one of my PhD students, now postdoc, made this molecule. Still remember, he put it into aqueous solution and it became orange, yellow-orange, which basically we thought, oh no, it simply died. So we added a TCO and realized, oh, wait a second, it decolorized. So maybe there is some chemistry going on. And in fact, it later turned out that it is, and that even the elimination works very well. So we achieved basically complete release in 90 minutes. This is how long the release took, which is basically due to a competition of these two OH groups, which means we cannot really control that only the right tautomerization is going. So we were very pleased with this, as this is sort of the first bis-pyridyl TC that can really be used for bioorthogonal bond cleavage. And we could move up the click rate to 1,300, which brought us quite close to this green shape here. And to finish, there's one last thing that I want to mention. So thinking of this tautomerization process, we basically then thought, okay, can we control this via the TCO structure? And inspired by Milan Fraber's group, we thought, okay, can we add this OH group here at the TCO scaffold that will later control the tautomerization, form the right tautomer, and eventually lead to release. However, in the meantime, we realized that even with pyridyl, even if we get the right tautomer, there is still no release happening. Which means, okay, if we achieve this, we might still end up with a non-releasing system. So, however, Walter, PhD group also, PhD student also in my group, he was already, let's say, too far to quit. So he made this molecule and we thought, okay, then let's test it. So he clicked it with the bis-pyridyl tetracin and we thought, okay, yep, we should get the right tautomer and maybe we end up with a compound so that we can study what's going on here. And he came back to my office and said, no, it's actually done. So we have complete release. And I was like, okay, how fast? And he said, no, the first sample. We're like, oh no, that's impossible. Because we know that, right, if this clicks and we get the right tautomer, the 1,4 elimination cannot happen. And in fact, this is what he showed. There is no 1,4 elimination, as what's going on here is basically a new mechanism. It's as we call it, an intramolecular cyclization. And due to this intramolecular control of tautomerization and elimination, we call this molecule the ITCO, which with bis-pyridyl, just to put this into perspective, has a second order rate constant of 10,000. The release is instantaneous, it's complete. And as we know now, it's also very, very stable. So just an example, 3 micromolar plus 6 means it's complete in less than 5 minutes. And we finally ended up here in this green area, which we are actually quite happy about. And to wrap this up, I remember the cube. I said there's a flaw that bothered me from the beginning. Yes, there's of course a corner hidden here in the back. And I'm happy basically to say that this is sort of our new release product here, which is formed from this tautomer here. It is part of the release phase. So we finally, let's say, found the exit from the cube, which is basically the back door that has been hidden so far. And with that, I'd like to thank all the people who really do the work here, all my team members, of course, also the entire group, my colleagues, and also the young students joining us. Special thanks also to Jonathan Carlson, a genius when it comes to chemistry, biology, and medicine. Very glad to have him as a friend and collaborator. Of course, also thanks for the funding, and finally to you for your kind attention, and sorry for going over time.
Okay, thank you very much, Hannes. I see that the molecules that are behind you you even have bigger ones of them, like you showed here on the pictures.
Yeah, but those are made out of plastic. I need to admit.
Okay. Well, thank you very much for sharing this, I would say, intricate details of release in those cycle addition reactions. Meanwhile, some questions have come up. And so one question that I see is, are your bis heteroaryl tetrazenes, are they planar molecules?
That's a very good question, because that's sort of part of the hypothesis also when it comes to deprotonation, why those molecules are no longer pink. In fact, we cannot say whether these compounds are still planar, but computational investigations would suggest no. But there will hopefully also another paper soon come out that will show that a bispyridal tetrazine, so no OH, is not planar either. So yeah, basically hard to answer the question.
And another question is from a person who considers himself a complete outsider in the field. Would it be easier to make the diacid of the tetrazine instead of making the allyl-diol derivative?
Yeah, I skipped that part and still went over time. So we made the diacid already, that was already included in the first paper. That achieves complete release. It's similar to the, let's say, yellow tetrazine, but the click rate is really low. So it was the first TZ that achieved complete release, but the click rate was, I guess, less than 10 per molar per second.
Yeah, I guess that answered the question. Perhaps one last question from my side. So of course, there are advantages to being able to release compounds, being able to release drugs at will in a living system. On the other hand, it requires two APIs that need to be approved by FDA or EMA or whatever. So how do you look at that?
That's our enemy question, always. And I can only cite or quote basically Mark Robillard, and also from their papers. So if you would consider the ADC approach, and you would have, for example, 10 different ADCs, all these ADCs could be, if you're lucky, cleaved with the same scissors at the end. Which means for 10 drugs, it wouldn't mean 20 compounds, it's 11.
That would, that would certainly help. And on the other hand, or also to support that this is a viable approach. I mean, like you mentioned, there is currently an ADC in clinical trials, which also shows that it must be a feasible approach indeed. Yeah.
And I should also say, right, just it's always costs and tox study and translation. But at first, from a scientific point of view, I think what's also important is the question on whether we can do this, right? If you would go back 40 years in time and compare to what we have right now, then you would need to cancel many things because it didn't seem feasible back then.
Yeah. And then maybe a last one that comes up that says, you made no comment on the difficulty of the synthesis of tetrazenes. Could you use different heterocycles used for click chemistry to apply the release you want?
Yes, I made no comment on how tricky it is to prepare tetrazenes, or how tricky it is to prepare transcyclooctanes. You know that even better than I do. But yes, definitely no time for that. Other heterocycles, there are triazenes out there. But as far as I can tell, and as far as I know, there is no other heterocycle that would achieve similar rates than tetrazenes. And yes, the synthesis is not easy, but most of them, at least most of them, are pink. So you saw the column, it's usually, I'd say easy. People here would kill me, sorry, to get those.
Yeah. Okay. Thank you very much. Well, synthetic difficult it could also be an opportunity, because that still requires that we come up with better methods.
Oh,
those compounds.
Several new methods out there, mainly from Joe Fox, that are really great, that made our life much easier.
Okay, thank you very much for presenting, Hannes.
Thank you.
Then we will move on to a brief intermezzo, which will be presented by Karen Muirhead of Thieme, who will give a short demo on the eSOS system content. So Karen, the floor is yours.
Thank you, Floris, and thank you, Hannes, for the interesting talk. Science of Synthesis is the largest collection of organic synthetic methods available worldwide, and it gives you access to more than 20,000 reliable synthetic methods. It's written by chemists who are experts in the area that they're writing about. And so the reactions and the structures that they've selected give you the best reactions. As it's a full text resource, the advantages and drawbacks of different approaches have been discussed, and the authors have added their unique insights into the science. Science of Synthesis is unique in that way that it provides critical reviews on synthetic methodology and organic and organometallic chemistry. So ultimately, this can save you time when you're planning a synthesis by giving you ideas for how to tackle a difficult step in your synthesis, or maybe you're just looking for a good and reliable method for a particular transformation. If I scroll down here, you'll see that the core content is organized systematically and logically based on the product, so specifically the main functional group that you want to make, and it's structured and text searchable online, so text searchable here and structure searchable there. As it's a series of reviews, it can be particularly useful also in getting background information when you're starting a new project or a new stage in your project, and it's also useful for teaching and for consulting. To complement the core product-based structure, the trends and innovation here in the reference library show they focus on the methodology and important techniques and technologies, and this is where we find the click chemistry volume. Just click in there. You'll see here introductory information on the more traditional click chemistry and its applications, and just here, see what sort of things that are in there. The reference library topics are also mainly focused on synthesis, but not exclusively. They give a lot of background and introductory information on principles and practices. For example, if I just go into the tetrazine section from Professor Mikula here, there's lots of information on the reactivity and stability of the tetrazines, like he showed before in his talk. It gives Science of Synthesis presents chemistry in context. So each full text article has the same basic layout, and that makes SOS user-friendly. So as well as discussion of synthetic methods, the scope and limitation of reactions is discussed, and where appropriate, there's also preparative details for the reactions. So I'll just show that in from Professor Moses here. The discussion and then appropriate experimental procedures. Links actually to both Professor Mikula and Professor Moses's chapters have been put in the chat, and these chapters are available for free until March the 8th. And I'll now hand back over to Floris and to Professor Moses for his talk.